Good morning, everyone. It is my great pleasure to welcome you here. My name is Tomas, and it is my privilege here to have two expert toxicologists together with me, Jarlath and Adam. We are actually sitting in one room. It's 12 December, 10 o'clock here in Prague, from where we're broadcasting. And I will give you one more minute for everybody to join. Get yourself comfortable. I'm very happy to see many familiar names in the audience. Hi, Gavin, our frequent visitor of the webinars. I can see Katerina, Martina, Olivera. Good morning to everyone. Before we start, I would like to emphasize what we are, trying to encourage in our webinars is live interaction and exchange of views, exchange of perspectives. So besides the Q&As that you can use, of course, feel free to raise your hand, and we will try to put you live on the webinar if you have any questions that you would like to discuss or any comments or experience that you would like to share. The title that we chose for today's webinar is New CLP Hazard Classes, How Deep Do I Need to Go? So what we want to cover today are different levels of assessment that you can or should do in different situations. So house rules first. Just a second. Yes. What you're gonna get from this webinar is the recording, the presentation slides, and our popular Q&A sheet. So you don't need to take any notes, and we will make sure to answer any questions that you might have. If we are not able to cover them live during the webinar, we will get back to them and send you the answers in writing, and we will circulate the Q&A sheet to everybody. We will not be really covering the basics of the new CLP Hazard classes because we did that in our previous webinars. Feel free to watch them. They are online, publicly available in our YouTube channel. And of course, if you have any further questions with REACH, CLP, or chemical regulations in general, no matter if it's EU, UK, now also Ukraine, which we are covering, feel free to contact us and turn to us with any questions you might have. And we have a specifically dedicated page to the new CLP Hazard classes. Originally, it was focused more on the endocrine disruption. You will probably learn in this webinar why EDs are the biggest chunk, we think. And with that, I would like to introduce our toxicologists here today. Jarlath, welcome and thank you for coming. It is a great pleasure to have you here also in person.
Thank you so much.
I will also add my bit in this talk, in this webinar, and then we have Adam, who will not have a presentation himself today, but he is here with us to answer any questions and join the discussion. Before we start with Jarlath's presentation, I would like to ask you, because I saw that we have a lot of consultants, a lot of experts, a lot of expertise in the audience. So if you can share with us what is your experience with ED assessment, PMT assessment, PBT assessment, it will give us some picture of how deep should we go in our presentations and in our discussions. So the poll is now on the screen. If you can cast your vote, we will show the results directly on the screen right away. I can see the votes coming in, and it's very interesting distribution, very interesting proportion. I will give you 10 more seconds. So we are happy that we can share some of our experience with those who came for the reason to learn about the field where they are new. But I can also see that there's some experience with the classifications and ED, PMT, PBT assessments. What is quite interesting to me on this poll is that ED and PMT is as it is new, there is very, very little experience. Of course, also with us, that's something new, but we are gaining experience fast, I believe. PBT assessment, there is some experience because that's a well-established category itself. So without further delay, I would like to ask Jarlath to start with how deep can you or should you dig when you are doing your assessments?
Thank you, Tomas. Share my screen. Okay, good morning, everybody. Hello, thank you. My name is Jarlath. I'm a toxicologist. I've been working in this area for quite some time. Thank you to Tomas and to Adam, the Regardless team, for having me today. This is a follow-up from the previous two webinars where Adam and Alish went into quite a lot of detail on the legal and technical side, specifically related to the recent publication of the CLP guidance on endocrine disruption. But the purpose and the reason I'm here today is really to try and bring in a more practical approach. As in, you have an inventory of substances or mixtures, and where do you possibly begin in order to classify or to check whether some of these substances need to be classified for endocrine disruption? It's a difficult and complex area, but it can be broken down into a set of systematic steps, and I'll bring you through those today. Okay, I'm not going to go into a lot of detail on definitions because Adam has done this in previous webinars on the details. I'm keeping this very high level, but it's important again just to go back to the original definition of endocrine disruptor and pointing out the two words for me as a toxicologist that stand out are the adverse health effects and intact. So as a toxicologist, we're interested in adversity. We look for adverse effects across a whole range of systems. So what this definition is really saying is that you need to show there's some detrimental effect to an organism in vivo, and that needs to be translated to populations and backwards. So just to bring that into focus, the classification criteria for endocrine disruptor is really a three-step process. And the first step is looking for adversity. So that has to be your initial screening approach, is to check whether your substance or substances reveal any adverse effects or suggestions of adverse effects on the endocrine system. And if it's clear from that initial assessment that there's no adversity, then you don't need to go ahead and do mode of action or plausible links work. So your classification work is essentially finished if you can show that there's no adverse effects on the endocrine system. So the question is, how do you check whether there are adverse effects on the endocrine system or not? Well, because it's a three-stage criterion, and also going back to other guidances, and of course CLP guidance is important, but there are other guidances, including the ECHA guidance from 2017. But we also have earlier than that the OECD conceptual framework from which the EU guidance is largely derived. And from this, we have basically an assessment strategy for endocrine disruption classification that we can apply. So first of all, let's just check the usual data sources prescribed lists. Are your substances already listed by ECHA or in the plant protection regulations or the biocidal regulations as identified endocrine disruptors? If yes, then you're going to have to classify. In most cases, not. There are other lists, but a lot of the lists out there are lists of suggested or potential endocrine disruptors that have not been fully assessed. So take those with a pinch of salt. Always go back to the regulatory source. Okay, so you've checked your prescribed list, your substance is not on this. Assuming your substance is registered in the EU under REACH, then of course you can access the public dossier to already look at the endpoint data. And that's what's going to be most important, as I'll come on in the next slide. But here you're looking for any data from the toxicology, especially the in vivo toxicology and the environmental aquatic toxicity. If the chances are you won't have access to the original study reports, and that's okay because the REACH dossier contains the robust study summaries, which are supposed to be good synopses of those study reports. The experience tells us that it's not always the case that effects are properly reported in these robust study summaries, but they're improving all of the time. If you have questions, of course, as to whether such a study is correct or full, you can always go back to your supplier to ask them if they have updated their REACH dossier recently, which they should be doing regularly. Okay, there's also read across and categorization. So this is where we can assess groups of substances, and this is perhaps the most economic and efficient approach to doing endocrine screens. And I'll come to this a little later also. But it's also important to keep in mind that a lot of substances are metabolized either in the body or they're degraded in the environment, so that there will be metabolites. So it's not only the original substance that you want to assess, but also any potential metabolites. And it's important to keep that in mind because some of them can be quite important. Okay, so you have the REACH dossier data, which is readily available. The next point is to do some literature searching. And again, we'll show you how we would approach that. There are then other available endocrine disruptors databases, and I'll talk in particular about the US EPA system. And then there are QSAR, quantitative structure activity relationship models, which do lend themselves well to endocrine assessment, and I'll discuss a couple of those. So once you've gathered all of those data and you've systematically assessed it, then your first question is, do any of these data suggest adversity? If the answer is no, then you can write your weight of evidence and into a document to keep that on file, because that's your conclusion as to why the substance or substances do not need to be classified under CLP. If there is evidence of adversity, then you go to the mode of action approach, and that's where you do need some quite expertise in toxicology in order to make links, or in a lot of cases show that a particular endocrine mode of action is not related to the adversity and that there's no biologically plausible link. But I'm not going to cover those aspects today. Again, we're talking about the screening, so we're very much looking for adversity. Okay, so looking at your REACH dossier, how do you tell if the substance is causing any adversity to the endocrine system? Well, there are some tests, of course, that are better than others. And for human health, mammalian toxicology, we're mainly looking at in vivo repeat dose tests. And those that would look at organs that are relevant for the endocrine system. So, these are the typical endocrine organs. Some studies will do extensive organ and tissue investigations, some will not, but the ones you need to be looking for are clear. So the typical endocrine relevant organs and tissues like the pituitary gland, the thyroid, the sexual organs, the ovaries, testes, the thymus. Don't forget the kidney is an important endocrine organ also. So we also have, some tests now look for specific hormone levels. So that's the extended one generation test, for example. Important to look at sexual function and fertility that we see in reproductive toxicity and in developmental toxicity tests like, PNTT studies, for example. We look at developmental toxicity, any effects on reproduction or population level effects. And also when we're looking at environment, we're looking at fish and the potential effects of the substance on wildlife populations. But there are also other areas that are not specifically covered by existing tests, but where data may be reported as part of the clinical biochemistry or urinalysis of those studies. But essentially, all of these endpoints are selected based on the OECD conceptual framework. So this predates the EU CLP guidance and the ECHA FC guidance, and this is really what those are built on. And the OECD conceptual framework includes guidance on what tests are most relevant for looking at endocrine disrupting effects of substance. And it puts those into tiers. So there are 5 tiers of tests, with tier 5 being the highest quality, but also the most complex test that would look at the potential across multiple endocrine modalities. Remember these EATS modalities, endocrine EATS that Adam has been talking about in the past. So we're looking at those modes of action, and we see, for example, the extended one generation assay, which of course is very extensive and expensive test, does cover all of the EATS endpoints. So it's a high quality study for assessing. Reported in the REACH dossier. So you look at the various tests that are available in the REACH dossier, you compare to the guidance document, and it gives you a good impression of the quality, level of quality of data that you're using to assess. And those studies would be useful then for you to look in at all of these particular organs and to see if there was any pathology findings or any histopathology amongst these organs that would suggest some adverse effect on the endocrine system. Just a note that some of these organs also are subject to spontaneous lesions in some species of rats and mice. So you do need to be quite careful in assessing these in accordance to historical control data, etc. So although this is a systematic approach, you do need to have some kind of expert toxicology input at some cases to make sure that you're assessing the results correctly. Okay, so you've looked at the REACH dossier. The data may be good, they may not be, but there are other information sources that you can check, and probably the best among these is the US EPA Tox21 system that includes the so-called endocrine disruptor screening program that's been running for a number of years already. And this is a collection of high throughput assays that have been performed on, I think there are about 1.4 million substances in their database now, and some of them have been tested quite extensively. Including specifically in estrogen, androgen, thyroid, and steroid bioactivity. So there's a special section where they have collected all of the results of these ongoing screening tests to look for endocrine relevant endpoints. So this is a very, very useful. The data are a little complex, and it does again take some expertise in order to extract and to assess these data in the correct context. But this is a really useful source of data to add into your weight of evidence. Another data source are the quantitative structure activity relationships, the QSAR models. And as I mentioned, well, you know, where these models are quite useful for endocrine is that they're rather good predictors of receptor binding and receptor binding affinities. So the experience is that the existing QSAR models, and there's a lot of them still coming on stream, you know, in the last year in particular and in future, but there's a lot of new models coming on. And it's found that if your substance falls within the domain of that model, or whether it's the model is relevant for your substance, then the prediction is usually pretty good from experience. So we have the QSAR toolbox from OECD, which is rather, you know, top heavy. It's difficult to use. But there are other systems, such as the Danish QSAR database, which is a free open database of QSARs run by the Danish EPA. They give free access to that, and they have a growing number of endocrine models. And we also have in the EU the Vega QSAR toolbox, which is essentially the proving ground for the QSARs that end up in the OECD toolbox. And it's a much simpler system to use. So typically I would start with the Danish QSAR database and the Vega QSARs. Which will already give you access to a lot of good models with very good documentation that again adds to your weight of evidence. So, you know, at this stage, you've assessed 3 lines. You have the data in the REACH dossiers, the endocrine disruptor screening program, and your QSAR models. You already are probably in a good place to do a weight of evidence assessment, in that if looking at these data sources, you don't see any evidence of adversity, then you're in a pretty good position to conclude that your substance or substances are likely not endocrine disruptors. And if you have seen adversity, you already have some pointers as to where you need to continue the assessment, because if it's just thyroid adversity, then you know to look at the thyroid specifically. Moving on, there's also literature search. This is where we need to be comprehensive. If you're going to justify a non-classification, then you have to show that really you've checked all data sources. The literature search is quite specialized and has to be quite comprehensive, because you're looking not only for specific endocrine, but you're looking for a lot of effects that may be suggestive of endocrine effects in metabolism, in alterations to male and female reproductive function, and in the environment. So you need to do a quite widespread, throw the net quite wide. And in order to do this, Regardus has developed a series of queries specific to endocrine and related endocrine effects that can be used. And this is just an example of the kind of complexity that we see in these literature searches. But there's now a pretty well researched and stable set of search queries that can be used that would comprehensively cover all endocrine related endpoints. Now, in the substances that I've been reviewing, when we do the literature search, we often find thousands of hits. But in the end, very few of those will actually be relevant to the endocrine assessment. But at least you can show that you've done the background search to see whether there's any new data. Okay, just to briefly touch on the difference between substance category approaches and why you should be going for category assessments where possible. Because we're looking at structural alerts for endocrine, substance categories based on the structural similarity or the existence of common metabolites are generally good candidates for efficient and quick endocrine disruptor screens, like I've just discussed. So if you have a chemical category that's been codified within REACH using the read-across assessment framework, then you're in a good position to assess as a group. The common structural elements may be well covered in the existing QSAR models. And metabolism, as I mentioned, it's always important to consider metabolites. And a good example of this is, esters are generally hydrolyzed to a common carboxylate and a corresponding alcohol. So in such a case, the metabolism may be quite rapid. So the original substance is not hanging around long enough to potentially cause any endocrine effects, but its metabolites may be. So you need to look at the metabolites also. And as a case study, looking at alcohol metabolites, these are the short chain aliphatic alcohols. What we know about alcohol is that high dose exposure in humans can lead to endocrine effects and damage to the fetus. The question is whether we need to be assessing this as part of our endocrine classification investigations. Well, we look into a little more detail here. Yes, ethanol does cause teratogenic effects at high doses, but we have very little information on the low dose exposures. What we do know about high doses is that it's probably not an endocrine specific effect, but rather it's a more widespread pathology related to, ethanol poisoning. We also have a lot of data in the REACH dossiers for alcohols on methanol. So methanol was used as the read-across source for most of the alcohol studies in the dossiers. By looking at methanol, we also see that it causes teratogenicity, but it only does this at doses that will be toxic to the mother, and is seen to be exacerbated by folate deficiency. So what we're already seeing is that high-dose alcohol, yes, possibly effects on the endocrine system, but probably not an endocrine-specific effect. But when we do literature search, we find some more interesting information. For example, we see tertbutanol, which is a common metabolite of MTBE, which is a suggested endocrine disruptor. Tertbutanol has been tested in vitro along with other MTBE metabolites and was found not to be an androgen receptor binder as was originally thought. So tertbutanol can be ruled out as an endocrine disruptor. Similar for 2-ethylhexanol, is a DEHP metabolite. DEHP, again, another endocrine disruptor. But there are specific tests that looked at DEHP metabolites and found that 2-ethylhexanol also did not affect steroidogenesis. So you build up your evidence like that, and for alcohol metabolites, we can reasonably conclude that at low dose alcohol metabolites are not causing endocrine disruption. But the point is, we've done this, we've come to that conclusion in a systematic way. So we've looked at all of the data and we've codified it and put it into a report, and we keep that report on file for whenever the authorities, if ever, they come knocking to say, what did you do to classify your substances? And so while we looked, we applied the endocrine criteria. Number one, adversity. We found no adversity. Here's the documentation proof of this. There's your justification for non-classification. If you do find adversity, then you're moving into the MOA analysis with Adam. Okay, so moving quickly on to the conclusion as soon as possible. I'm not going to go deep into this, the application of endocrine criteria, but again, based on the screening approach, where you're just doing a systematic check of the existing data, if you're not seeing adversity, then you don't have to classify, and you don't have to move on to the more complex analysis. If you are seeing some adversity, and I keep saying this word adversity, but that's what the legislation requires. You look for adversity. If you don't see it, no classification. If you do, you move on to the mode of action analysis. And further, CLP does not require you to generate information, but there may be some new requirements in future. So always good to know your gaps and deficiencies as well, even if you're doing just a straight up weight of evidence assessment. There are suggested lines for when you can clearly say that a particular endocrine activity is sufficiently investigated, but as you can see, these are generally not standard information requirements in REACH. So this would require some specific testing, and often these data are not available. So you're coming to an expert judgment based on weight of evidence. Okay, so we looked, most of what I've said so far has been related to specific to human health, but there's, of course, there's also the endocrine assessment. And as we see, under CLP, endocrine and human health are kept separate. They're separate classifications. However, the classification for human health can inform whether a substance may be an environmental endocrine disruptor as well, particularly around the thyroid effects, because thyroid is well preserved in vertebrates, like fish. Fish also have thyroids. So if you're already classifying it based on thyroid effects, you can do that for environment as well. However, the experience is that most REACH dossiers, especially, they don't have very good quality aquatic toxicity data that would allow you to come to a firm conclusion on endocrine disruption in the environment. So again, you need some careful interpretation of the data. Often you may only have the mammalian data, and you may have some invertebrate studies that show effects on Daphnia or maybe their offspring. But that's often not enough to reach the bar for have you sufficiently investigated. But again, you don't have to generate new information at this point. It's just looking for your various lines of evidence and how they tie together. The environmental side of things also brings in the concept of potency, which basically means that if even there's a slight effect or slight suggestion of endocrine effects, then it probably will end up being classified, even if it's a very low potency endocrine disruptor. But again, for the environment, the issue is usually not sufficient or not good quality data in order to come to a firm conclusion. So it's good to assess human and environment together to come up with your justification. And in doing so, you've done a screen essentially of your substances without having to break the bank or to spend effort on a lot of things that you might not have needed to do. For example, if you're not seeing adversity and there's no MOA required, then you're in a good position. So hopefully that's been somewhat informative. Again, not comprehensive in the time allowed, but at least it's a systematic approach that you can apply. But as always, there are areas where you need expertise in order to properly weigh the evidence. And in those cases, you should try to consult with a toxicologist who will be able to tie those lines of evidence together for you. Okay, I'll leave it there. There may be some questions that I'll take later. Thanks.
Thanks a lot, Jarlath. It was super useful, at least in my eyes. And, seeing the questions that we are getting, I think it resonates very well with the concerns or questions of the audience. There's one particular question I would like to take now before even opening the Q&A session, because it relates to what you just said in your last sentence. Jelena is asking, are EHS consultants qualified to do ED assessment, or is this task explicitly reserved for ecotoxicologists? It seems to Jelena that ED assessments require specialized knowledge in ecotoxicology. And I can see also Adam taking a microphone. So who wants to start?
Well, I'll start. What I was discussing was the screening approach, and you have to do something like that in the first instance in order to get a feel for where you are in terms of, do I need to classify or not? I believe that for the screening approach, you don't need to be an expert ecotoxicologist, but when you're getting into the mode of action, the side of things, if you're doing a deep dive assessment where there may be new data required, then you're in Adam territory.
Yeah, so I would add that the data set is assessed as a whole. So you have, there are some triggers from the ecotoxicology studies, you have some triggers from the mammalian rodent studies, which are actually non-target mammalian species also used in ecotoxicology. So the borders here between toxicology and ecotoxicology are not so strong. I think there needs to be understanding both of the rodent toxicology and both of the amphibian fish toxicology, ecotoxicology. The endocrine is very preserved. So basically the same QSARs are used for ecotox. It needs to be look at as a whole data set. But then when you are doing the decision, you are doing two decisions. One which will be human endocrine, and then second decision will be on environmental endocrine, because in environment you are looking on the effect on the population, whereas for human health you are just looking for adverse effect within the animals.
Okay, thank you. I think we got the answer. If you want to talk to Adam, please reach out to us. If you let a scientist speak, he can talk weeks about this. But the bottom line is that for the basic activities, basic items, you don't need expertise, to go through the existing lists of EDs. Even I could probably do that, I guess. But when you go deeper, you really need to assess the data, and that requires expertise in toxicology or ecotoxicology, right?
I think the point is that you can take a systematic approach, which means you can bring in the expertise at a later stage when it's required, but the initial screening is usually pretty straightforward.
Thank you. Thank you. Makes sense. Okay, we will get to more questions after my presentation, which I will go quite quickly through. So I'll start sharing the screen. Okay. So just a recap of the timeline. We covered this in our past webinars, but to bring it back, that time to act is now for some of the actors. So if you have an obligation to classify substances, you should start quickly. Mixtures still have some time. That brings me to a question to you again. How many chemicals do you think you will need to assess for new CLP hazard classes? And the reason I'm bringing this question up is, of course, that it can get lengthy. To do the rapid screening, that's not so much of a hustle, but still, if you get dozens of substances, unless you can use the category approach. If you have some group of substances, you are facing a serious challenge or serious workload. And then, of course, if you have multiple substances from completely different groups or of completely different chemistry, you are potentially up for a very lengthy job. So let's see how it goes. I'll give you 10 more seconds. Oh, this is interesting. And the numbers still keep piling up. Thank you for participating. It's really interesting to see the numbers. We have 47, 49. Let's wait for 50 answers. One more answer to come in. Okay, everybody probably cast. Yeah, we got it, 50. S.r.o, can you please share it? Okay, so it seems pretty evenly distributed. Some of you, the lucky of you, I would say, do not need to assess anything. Some of you will need to assess only a few. Some of you are facing dozens of substances, and most of you, the highest number, a lot. Interesting to see the mixtures as well. There, the statistics seem to be skewed a little bit towards a lot, which I would say was expected. Thank you. So thanks to Jarlath, you saw how you can systematically approach it to knock out maybe some of the substances which you will not really need to assess in detail. So we divided this into 3 levels of assessment. Tier 1, which we call rapid assessment, should give you early indicators for classification and knock out clear no classification cases. So if you run the QSARs, if you search for structural alerts, check the existing lists of EDs, that should already give you some good start. But for the classification itself, you will definitely need to go to the REACH data and to the studies, at least the robust study summaries, which may themselves not be sufficient, but it's a starting point definitely. And you will need to assess available information in literature. The big question here is, how systematic and how thorough you should be with the systematic review and the literature review, because of course, there you can spend months going through all the different scientific publications, which you will definitely need to do when you go to tier 3, which we call full detailed assessment, where you really try to collect everything that is out there and assess everything that is out there. And that's definitely what you need to do when you face some disputes, some litigation, court cases, or public consultations during harmonized classification proposals, or simply if you are, facing some disputes with your suppliers or customers who think that the classification should be different for any reason. So this is what it takes. I will not spend too much time on it because I think Jarlath covered it clearly. But I guess you might be interested in having a sense of how much would it cost, if you hire a consultant to do this, some indication of how costly this is. So the way we calculated this is that the rapid screening should be somewhere in the range of 2,500 euros. Of course, if you have similar substances, use the grouping approach, it can be a little less. To do the minimum you need for classification, so that means also going through the REACH data, typically comes to 5,000 to 8,000 euro. But of course, the range can be completely different depending on the volume of data, for heavily populated substances with a lot of data. This can be more for substances with either no data or clear no classification cases, or on the other hand, clear classification cases where you can instantly see it's an endocrine disruptor. It can be less. And full detailed assessment, well, that's a big that's a big question mark, which really depends on the volume of data and the nature of the substance. But it typically starts at 20,000, somewhere in the range of 50, 60. If it's, I would say, more straightforward case, but then if you are facing a complex substance and complex case, we know that this can take a year or two to really go through all the data and exchange views with different experts. So I already indicated which tier you should go to in different scenarios, different cases. A rapid screening, that's your starting point, especially if you have a large portfolio of substances. It should help you to assess your workload in future and knock out a few candidates which are clear no classification cases or clear classification cases on the other hand. Tier 2 is for anyone who has the obligation to classify substance or mixture. And Tier 3 in cases where you are facing any disputes or different opinions with your business partners or other stakeholders. So we are ready to help with all these tiers, including, for example, biocidal products and PPPs, where Adam has experience, extensive experience, I would say. I don't know if that's good for you or bad for you. And please don't hesitate to contact us if you need any more information. We will definitely be doing more webinars on this. We want to keep on, discussing the latest news and also exchanging views as the experience grows in the community. I would probably kick off the Q&As with a question to our panelists, to Adam and Jarlath. What do you find most challenging in classification of EDs, PMTs, and PBTs?
Well, for me, there's a difference between EG and PMT, PBTs, where PBT, PMT are very criteria focused, on numerical values, whereas endocrine does require a broader approach from an expert point of view, because there's a lot of different factors to consider when coming to a conclusion. I think the guidance is not clear in some parts, the CLP guidance, but it's planned to improve over time as more people get experience of actually doing these classifications. So, I mean, there's very little experience out there at the moment. I mean, apart from the screening work that I've been doing, I haven't led to a situation where I'm formally classifying for endocrine disruptor effects so far. PMT is different. I mean, already we can identify substances that are not formally classified as PMT, but probably will, because the criteria are clearly met.
Thank you. And your view, Adam, your perception of this?
Yeah, it's I think the point that actually the guidance is kind of first round. So as the states and ECHA were not able to decide or to say what are the category 2 no classification, category 1 examples are there, but in the guidance they could imagine just completely pure imaginary substance, a very clear example of a category 2 and very clear example of no classification. And apparently there is no agreement between the ECHA and member states what is what. So then it's quite difficult to actually for us to assess when there is no clear boundaries. So maybe the category 2 came from the ECHA guidance, where the authorities were not able to decide if it is endocrine disruptor, it is not endocrine disruptor. So it's kind of when they want to classify, but they don't have evidence. And I don't know. So for me, either it's endocrine disruptor or not. And again, it should be risk nor hazard.
Okay, let's not get into that kind of discussion. We can leave this for some other occasion. The way the Q&As work in our webinars is that we try to prioritize anybody who wants to ask live. So raise your hand if you want to ask live, and we will make sure to answer your question immediately. Then we prioritize questions in the Q&As panel, and then we have some questions from you from your registration forms. So we will try to get to those, but we will first answer those live and in the Q&As panel. Starting light, well, hopefully light. Question from Barbara: what would be an example of a clear no classification case?
Well, maybe I can answer that because I've dealt with such a case recently with a category of substances that I was assessing. They were extensively documented. All of the REACH standard tests were in place. Some read across, but mostly as a category of substances, they were well represented by data. As I've just gone through all of the REACH dossier data, no evidence for any adverse effects. I mean, remarkable for the lack of systemic toxicity, but specifically on the endocrine or reproductive organs or any of the circulating hormones, no evidence of adversity whatsoever. All of the QSAR screens came out as inactive, and the endocrine disruptor screening program results all showed no bioactivity and no non-specificity. That is that the results were deemed to be valid and relevant for those substances. Literature search showed very few papers, but none of those indicated any issue. The only potential issue was with the alcohol metabolites, as I've mentioned, because these substances were esters. So as not only did I look at the esters in detail, but also the alcohols, and there was a very clear case of no classification required and therefore no MOA needed.
So maybe addition. Hello, Barbara. So some rich chemicals are not so toxic as plant protection substances, which are kind of designed to be bioactive very often. So there are cases where there's no classification, and also second. It's maybe not clear no classification, but in REACH there is no obligation for data. I think it's also kind of answering the previous question of you, like are there some indicators or expectation for data sufficiency when no additional testing can be triggered? So there is no additional testing in REACH. So if the data set is not full and there are no data saying that it's a endocrine disruptor, even though there are missing important studies, it's no classification. So up till there will be the update of the REACH where there will be requests for data. Also, no classification is done in cases that there are no data available, and the data which are available are not giving you some indication of ED.
Thank you for pointing this out. I think that's a very important aspect that, currently we are talking about CLP, which the mechanism is clear. Assess whatever is available and no need to generate new data. But that's likely to change in near future with the REACH review or with new update of REACH. It's quite likely that new information requirements will be brought up specifically for EDs, maybe PMTs as well.
Well, I see one question from Barry. Touches on this point about the availability of data. Yeah, you've pointed out the CLP does not require new testing. It's clear that there will often be gaps in data. There's never an ideal situation, and some of the endocrine modalities require specific tests that are not part of the REACH testing standard testing requirements. So, but that's where, unfortunately, toxicologists have to deal with such uncertainty on a day-to-day basis. There's never a perfect dossier, there's never a perfect amount of information. But you do have to, as much as possible, draw in the different lines of evidence until you can assure yourself that either, yes, my judgment on a no classification is can be defended, and you document that, Or where you yourself are uncertainty, you can identify the gaps for your clients and say, well, my advice to you is you can consider doing such a test, even though it's not required, but if you really want to be 100% satisfied. But the case in reality is people want some kind of assurance on a weight of evidence. Yes, point out the gaps, but they want something on file that they can discuss with the authorities if ever required. So just specifically about Barry, because I have a feeling Barry is probably a toxicologist. Also, Barbara, am I guessing?
Yeah.
Oh, okay, yes.
Sentient endocrine expert.
Yeah. So the nuclear receptor modulation that Barry talks about, yeah, this is just another example. Is where you have one piece of evidence among many, and you have to put it into context as a toxicologist. I mean, that's basically your job as a toxicologist, I would imagine, is doing that on a daily basis. So whether that will be frivolous or won't lead to classification is hard to say. But it's an expert judgment. It's a judgment call at the end of the day where you map out your uncertainties.
And what about Barry's point that, why starting with in silico data and it resonates also with Barbara's note where she mentioned, what if you have some indicators of endocrine disruption in the in silico data or QSARs, and what then? And Barry makes a point that maybe it should be the other way around. Start in vivo with in vivo data, and then
well, of course,
augment it with in silico.
Yeah, of course, and that's what I showed in my presentation. Is you start looking at the in vivo data. You're looking for signs of adversity. You pick up all of those and you document them. If you look at how the ECHA and EFSA document does it, they have this Excel sheet, and you have your green boxes and your red boxes for a huge list of endpoints. So essentially, what I'm doing is that, but on a much simpler screening level, because that takes a huge amount of time to do for one substance. But yeah, you're going to find little pieces of evidence that don't fit the preconceived map. But again, as a toxicologist, that's where you to express your expertise and have confidence in that.
But if I'm not mistaken, the point you were making in your presentation is that either if you don't have toxicology or ecotoxicology expertise, or when you have a big list of chemicals that you need to assess, it's probably more pragmatic to start with the screening to prioritize where to start and which substances to
okay. Even the screening level, you're going to require a toxicologist at some stage just to check, but the collection of the data shouldn't require a toxicologist. So you can collect the data and have it ready for somebody to assess.
Yeah, thank you.
Sorry, I don't know if that answered your question, Barry, but
well, I saw that some of the questions are really quite technical and an expert, and we need to go to them, I think, in writing. I don't think we will have time to cover them. But one area where we haven't really, which we haven't really touched yet, is downstream users and mixtures. So what can you say to that?
Well, from my perspective, I was looking at assessment of substances. For mixtures, for me, it's clear. If you have a mixture, then you need to contact your supplier and ask them to do the work to make sure that they're compliant with the law so that you can be. And that's how the whole system is supposed to work. If the supplier is giving you pushback, you just tell them that you're required by law, by compliance. And if they're having problems, then get them to contact Tomas.
Yeah. Okay, I maybe again added that this supplying companies should give you the data. Be sure that you have updated data sheets. Be sure that they know what they are talking about, because at the end, the companies putting on the market the mixture are responsible for the mixture. So they can say like, we rely on our suppliers, but maybe the suppliers, they need to be sure that the suppliers know what they are talking about. And also some of the mixtures, maybe some companies can underestimate, and you would like to be sure ahead that in your mixture there is no endocrine disruptor. So good examples could be even the plant protection products, maybe. So you need to check ahead that you don't have any of your rich substances added as additive for whatever reason into the active substance. that it's not endocrine disruptor. You don't want to have the product classified as endocrine disruptor because of some rich substance. And that takes time to exchange the substance in plant protection product. Or also, if you have toys or anything which you want to be really sure that there are no endocrine disruptors, or cosmetics, some perfumes or something, it's better to check ahead for yourself, to look for alternatives. Even you find out that in the mixture is endocrine disruptor, you don't want it there, based on regulation of cosmetics, for instance, but also for customers, you don't want to have some coverage in media, and also for yourself, you don't want to sell your customers some endocrine disruptor. So I think there is also the point of responsibility for your products.
Thank you. We are running short on time. I would love to keep on with this discussion, but I think it's not really fair towards those who have already plans and some follow-up meetings. So one last question from Barre, bringing up again the endocrine disruptors category 2, which we are getting constantly also in the previous webinars. Barre is asking, I mean, what does it tell the public if it's category 2? Does it mean that it's an unnecessary fear, but no clear proof? Or should the public be concerned whenever something is category 2 classified? I know it's a tough one, but I like the public,
Well, I think this has been a big subject of debate between the member states, so they themselves can't agree on this. So this is something that will have to be challenged, and the RAC, the ECHA Risk Assessment Committee, is going to have to make some kind of threshold or cutoff argument here, in my view.
Yeah, I think the categories have sense for carcinogens. They have sense for reprotoxic substances. It's quite clear how this is done. But for endocrine disruptors, I would say the ECHA guidance was better in this. Either you can say it's endocrine disruptor, or you cannot say it's endocrine disruptor. If you have the evidence, you say it is. If there is not enough evidence, I think it's sending to customers some message which, with the fear of chemical substances, it's kind of damaging, but maybe not really rational, because there is not evidence. And also the regulations are not prepared for category 2. It's not in the reach. It's not in the any regulation. There is not thinking about category 2. So it's kind of put there in CLP without really link to anything else. So it's big unclarity, which can raise worries in between the people like customers, but also in between the scientific community, because we don't know what it should be.
Thank you. I was just made aware that the webinar is officially announced to end at 11:30, so we still have 30 minutes, which I'm really happy to, and sorry for my mistake. But of course, if there is anybody who was counting on ending at 11, please let us know, and we will try to take it into account so you don't miss anything. But in any case, you will have the recording available, so you can watch it later. And I think we definitely need to do a more in-depth discussion in some future webinars once there is more experience with the classifications, specifically about this category 2, because it keeps popping up and the questions are coming from all levels. So we definitely need to go back to that. Let's take a look at some further questions. What about the non-EATS mediated endocrine disruptors? I saw one question around that. Yeah. So I will read it out loud for everybody. For non-EATS modalities, is there any feedback from ECHA if changes within homeostatic range will be considered as non-ED? For example, blood glucose levels, insulin, satiety hormones such as leptin. Would that indicate a threshold for non-EATS mediated endocrine disruption?
Yeah, I don't know what ECHA are thinking here, or whether they're going to make such an approach. I think it will be very difficult because if you're talking about going outside of homeostatic ranges, the question, again, as a toxicologist, I want to know where's the adversity? And that's clearly part of the endocrine criteria. You're looking for adverse effects. So are the blood glucose levels, are they way off? And is that indicative of an adverse effect? Or, I think it would be very, very difficult to justify dealing within the set ranges. I mean, if there's a clear adverse effect, then it's an adverse effect. But if you're just talking about a slight fluctuation in homeostasis, then probably not, because we're all living things. We're living, breathing, homeostasis changing all the time. So yeah, I would see that as a non-starter myself, but maybe, who knows? It's ECHA. Who knows what they'll do?
Anything Adam would like to add?
So yeah, no need to. I don't know if you saw the presentations from ECHA, and in the presentation from ECHA also the guidance. It's they are very proactively seeking any indications of endocrine disruption. The whole guidance is written like how to identify endocrine disruptors. So that's the point, to identify as much as possible, basically. So I think it has a very good chance to be. There are a lot of false positive substances in this approach. But at the end, it's expert judgment, weight of evidence. So I think somehow this needs to be assessed with best available knowledge and information from data. If this is somehow endocrine disruptor or not. So I think the borders are really not clear there, and it's actually also what ECHA is saying, that we will see a track what cases we will have, and then based on the experience gained in Iraq, we will further work on the guidance. It doesn't help too much. It's what they are doing.
So, as usual, the popular learning by doing approach.
Yeah.
Okay, we all need to do that, I think. Which brings me to a question of my own. You mentioned the ECHA guidance, you mentioned the ECHA webinar. Was there anything surprising for you in the recently published new CLP guidance on new hazard classes?
Yes, many.
Name a few or the biggest ones.
I do endocrine assessments, but I wasn't involved in the endocrine guidance. I did have some input into the guidance on mobility for PMT. And honestly, I wasn't happy with the way that guidance came out. I think it's very restrictive. I don't think it's very scientific. I think it's actually anti-scientific in a way. So yeah, but as Adam said, the guidance has to be considered a living document. I think experience is going to lead to inevitable changes as we go down the road.
Okay. Well, you are always the second one to comment. Maybe next time we start with you, Adam. But do you have anything to add?
Yeah, I think this was good. Yeah, we can go into great details here, but it's Charlotte. Yeah.
So as a scientist, your heart is broken with the new ECHA guidance.
Yeah. My scientific heart is broken from the first criteria on ECHA EFSA, where they said it's hazard, whereas it should be, because here you will have substances active in doses like 2 grams per kilogram when the livers get like overwhelmed, and then this will have effect on thyroid, you know, and that's no possibility in real life to really happen to anyone. And these substances will be classified endocrine disruptors, although the people would not be even able to eat so much every day, and it's just saying that something is hazardous where there is actually no risk coming from this substance. So I think it can lead to overusing of the hazard symbols, and then when public sees the hazard everywhere, then they stop
being effective.
Careful about the substances, and then they can take some, I don't know, some acid. Look out, there are again these signs which are everywhere and can cause damage. Actually, the over warnings,
over labeling. Yeah, good point. Good point. Thank you. And let's see how the policymakers approach this in future. But coming back to the questions from the audience. First of all, is there anybody who would like to ask live or share any experience, share any thoughts? If not at the moment, then I will start sharing the screen again and put on the screen some of the questions which came already during the registrations. So Marit was asking about any estimation how much the ED assessment will cost. I think I covered that in my presentation. Depending on the volume of data and the level of detail you need to go to, we tried to differentiate this into different tiers. So you got a cost estimate. We covered the question of downstream users and mixtures, classification of mixtures. Polymers. Is there anything specific to mention about mobility, bioaccumulation, or endocrine disruption when it comes to polymers?
Okay, well, I mean, first of all, polymers currently not subject to REACH registration, but remember, polymers are subject to CLP requirements. So you do need to ensure that polymers are classified. The reality is most polymers are not classified because people seemingly assume that they're not subject to CLP. Whether polymers are endocrine, that's a difficult one because polymers are such a broad range of structures. I think they would lend themselves well to, you know, structure activity relationship of not the whole polymer, but the structural entities that could make up that. But you may go back, you'll be looking at the monomeric data, the data for the polymers again. It's tricky. Polymers regarding mobility, I can't see them being very mobile substances. But again, it's hard to make a general claim because it's such a wide group. Yeah. Interesting question.
Well, if I'm not mistaken, the Woods report about polymers talks something about something like 200,000 different polymers on the EU market.
Yeah.
So it's hard to generalize. I agree, but I think the key message is that you need to classify, and of course, looking for structural alerts can help.
Yeah. Well, I mean, you know, polymers will be subject to registration. That's the intention, and in order to do that, they will have, you know, standard information requirements for polymers as well. The question is, will those information requirements be tailored to polymers, or will they just try to adapt what the current standard information requirements are? That would be the least favorite option because, you know, polymers, that can be better represented in tests. Yeah.
Well, I would not take a guess which way the future regulations of polymers would go, but I guess it will start moving again after the REACH review. Matthew is asking, looking for clarification in the coming into force of these new classifications, which tests and how to conclude. So Jarlath, you presented some testing methods. You spent quite some time on explaining the whole weight of evidence approach. Anything to add to this?
Well, of course, reclassification, CLP doesn't require you to generate data. So we've mentioned this point. I only touched upon the available tests. The OECD guidance document 150 is the one to look at for the complete range of tests. As you'll see, many of them are not even within the REACH standard information requirements. So you are looking at, if you want to do a comprehensive endocrine assessment, you are looking at doing probably the need to generate more data, but you have to balance that against the requirements of CLP, which as you don't. So you're doing a weight of evidence expert judgment. The question is what happens in the next REACH update? Will they include new specific tests for endocrine and try to really close it off that way? That's the question. Even I don't know.
Yeah, again, some guesswork for future.
So maybe like as you saw, even the assessment of current data is actually quite time consuming and complex, very complex. So although there is no requirement for studies or tests in CLP, the requirement just to screen the literature to go through all the existing data, it's quite to do the QSAR. So it's level 1, maybe 0. It's some work needed now, and when there will be a REACH update, if there will be a REACH update, then there will be this test, and we can expect maybe similar data set as in biocides plant protection, which is really huge, especially for the substances over 1,000 tons.
Yeah. That's quite likely in my eyes.
So it might be really expensive for these big substances, but the substances, they are produced in high numbers, but sometimes they are produced like with revenue for companies which are not so high because there is a lot of competition, and if it can even happen that for substances which are really important for us, that it will not have economic sense for companies to continue the production. So
yeah,
it's like, and also for the small substances, like from small manufacturers and so on, it will be like really difficult for them in monetary, I think, because if there will be strict requirements for the data to be generated and for tests, a lot of companies will just stop because it will not have sense to continue, and then of course there will be some imports and yeah.
Well, thank you. Without doubt, there will be huge impact on the market with these new CLP classes. Let's just hope that the socio-economic analysis before introducing these new CLP classes was really thorough and took into account all these impacts and aspects. Which brings us also to some further regulations in future. Daniel is asking, well, we spent quite some time how to determine what is an endocrine disruptor or different hazard classes. We also covered this in our previous webinars. But what about upcoming regulations for EDs and PMTs in future, and maybe not only in Europe, but also elsewhere? Are you seeing any indicators of where the rest of the world might go with this?
Well, clearly Europe is leading the charge and has been already for a number of years, and you don't see other regulations picking up. So whereas Europe requires formal assessment now, in the US and Japan and Canada, for example, they have a more laid-back approach, as in they may have some prescribed lists of existing endocrine substances, and well, you do a screen, and if there's suggestions to a screen, then you go with this. The question is, CLP is not aligned with GHS because of the whole endocrine, and that's something that needs to be resolved at GHS level. I know that there are some discussions going on. I know in the US they're constantly discussing about do they need to expand the endocrine assessment issue or not? Because at the basic level, what you never hear is there's still a discussion going on amongst toxicologists as to how relevant is this really? I mean, has Europe really overblown the risks of endocrine disruptors?
Yeah.
So the toxicology are split right down the middle of the
Well,
justification for it?
well, it is, publicly claimed by the Commission that alignment and harmonization with CLP is their high priority. So they are making efforts to introduce EDs on the GHS level. We will see how that goes.
So in the last ECHA presentation, there was a comment like when we can see that. So it was said that it is in process. That the commissions there are discussing this, and but they don't see it in the near coming years. It is alignment. So it's. ECHA and Europe is really pushing this, but on the other hand, some states maybe don't want this approach. So we will see how this is more, I would say, political now, the discussion.
Absolutely. Well, my personal guess would be that major markets like US, UK will not do anything before this is aligned or introduced into GHS. That's my personal guess. And of course, if it does get into GHS, then some follow-up actions by these countries and regulations might start, I think, but I don't expect any sooner. Interesting question from Monica. How can we use artificial intelligence to collect data for assessing chemicals for ED, PMT, and PBT properties? So how do you respond? And let's start with Adam first this time.
Okay, so when I was doing the first screens for the substances, I try, not the first, but like recently when the GPT-4 is kind of good, I tried to ask, like, tell me about this substance, is it endocrine disruptor? But it's just mixing a lot of information from different substances and saying like, so it cannot work like independently. On the other hand, when I was screening a lot of abstracts for looking into some theoretic effects, I could train the artificial intelligence to abstract me from the abstract the important things. So it was much faster for me to look for the triggers in the abstract to say if this article is relevant to inclusion for the thyroid modality or so on. So it can make the work faster, but it doesn't work as it would do the work instead of the toxicologist.
So what you're saying is that it can be useful, for example, for prioritization of literature data, of course, when you train it properly. So we have to give clear instructions and do a bit of work to make sure it works the way you want, but then it can prioritize really the relevant papers for you.
Yeah, I think there are potential applications, but there's a lot of work to do, you know. I'm still of the mindset it's garbage in, garbage out. And the little touch I've had with AI is not for search purposes, maybe some, you know, data collection and management, but it has to be at the moment very well managed by a human. I mean, because I just don't trust the AI models. They're generative, they're not helping us.
Well, that's what I wanted to point it out, that I think we are talking about generative AI, but let's not forget that, for example, QSARs are built with data machine techniques. So there it can help, and it is used for many, many years in drug discovery, drug development. So let's not confuse AI and AI, but generative AI, I fully agree that, we have to be really careful with.
Well, the reason I say that is unfortunately I have met researchers who are now quite heavily depending on AI. The question is, are you going to cede over all of your responsibility to a computer model?
Yeah,
you got to check the work. I mean, it still makes a lot of mistakes.
Yeah. Okay, thanks a lot. We are done with the questions from registration forms. I will check once again the Q&A panel. I can see there are still some questions, but which are really detailed, technical, and niche, I would say. So I would leave those for a written answer after the webinar. Is there anything else that you saw in the Q&A panel that struck your eyes and you think we should still address live?
Yeah, just again, going back, there was something from Barre. I'm sorry if I'm getting your name wrong, but you were talking about, lack of validated in vitro tests and the ability to conduct holistic experiences. I think the point is it would be desirable to have all of these tests done, but the reality is quite different. And that's, as a toxicologist, again, your day-to-day is dealing with data gaps and uncertainty, and how do you reconcile those, and can you come to a conclusion, and how confident you are in conclusion, and what are the uncertainties linked with that. So the reality is the data are never there the way you'd like them. You've got to do the legwork in order to try and bring it all together. And sometimes it comes all together and you can be convincing. If you can convince yourself and write that down, that's good. And sometimes you can't. And that's where you then have a data gap that you would like filled. But I just wanted to address that point. There is no perfect data set, and there never will be.
For me, the in silico model, I use it as a warning. So if there are like estrogen indications, then I look in the data for the really, like I set my mind for looking for the adversities in estrogen pathway, if there is something in vivo or not. So it's, I think, good indication. And also, if we go in vitro, I saw when the positive in vitro was not transferred into the positive in vivo. So even the in vitro model, so the substance really locked into the receptors. And it doesn't necessarily mean that there will be effects in vivo.
Mm-hmm.
Just the complexity of the body is very, very high. So
thank you. So that brings us nicely to the very beginning, where Jarlath mentioned that we are looking for adversity in an intact organism. I think that's the key message, one of the key messages from today. And getting to an end, some final words, take-home messages.
Good luck out there. You're going to need it. It's a lot of work coming our way, but we're just trying to make it more efficient. And that's the key thing. It's how do you manage this workload, because it's coming. Nothing can stop it now at this stage. And after this, you're going to have the PMT substances, and then you're going to have the polymers. So somebody asked the question, do we need more ecotoxicologists? And the answer is probably not. The world doesn't need them. But we need efficient ways to manage large amounts of data in order to make the assessments, cost effective.
I cannot agree more, definitely. Adam, your final words?
good luck.
Okay.
We will be able to help with the substances. So I like to work with this data set and look for this endocrine things in the data set. So happy to help to potential customers.
Thank you.
Also the current ones.
Thank you. As you can see, Adam and Jarlath are both very passionate about what they do. Thank you very much for this webinar. I enjoyed it very much. And I can only add that we definitely want to continue with this, digging deeper and looking at the new hazard classes from different perspectives. So if you have any topic that you think is important to cover, or any idea what our next webinars should be about, drop us a line. And thanks a lot and good luck. Enjoy your day and have happy Christmas.